sticky because they're looking for rocks to stick on in the environment. They will stick together. So we take advantage of that then. Once we break them up, we spread them onto the cedars, and we keep them in tanks. And because they're sticky, obviously they'll stick to the string. And we develop them up, and these are some of the little sporophytes that we have on our string. Whenever we're going to put them out at sea then, we have our logline site. Because Strangford Lough it's a very protected area. There are several different nature designations on it, and it's also very heavily used by a lot of people. There's a lot of pot fishing that happens from nephrops and lobsters and crabs and shrimp and everything else. But there's also quite a lot of recreational boating and yachts and things. So we've got a lot of stakeholders who are very kind of voiceful whenever they want to be about the impact that we're going to have. So what we've done is, instead of using normal anchors, we're using these eco anchors. And it's basically a huge mesh bag that's full of stone. And the mesh bag is non-toxic, and lots of tests have been done on it, it's fine. But it's filled with stone, and you can see there's lots of little kind of nooks and crannies and holes for marine life to come in, settle in. It's designed as a way to kind of promote an artificial reef habitat or an additional habitat on the seabed. So you're not actually taking any habitat away from things. So this is our site here. You can't really see very much. Northern Ireland, we tend to get a lot of grey weather, so it kind of works for us. If you look up around this area, can anyone see anything other than just sea? No? As you approach, especially on a sunny day, we've got these white buoys out. So you can't actually really see them from very far away, up to about maybe 100 metres getting close to the site. On a cloudy day, you can see them much easier from further away whenever it's bright. So these white buoys here, we've had no hassle with them so far. There's a town in the background. This is called Killilea, and the residents here haven't had any issues so far with our site that's there. Partly because it's still small, you only have a maximum of 6 long lines out at any one time, but generally it hasn't been too much of an issue. So this is the design that we're using. It's very basic. The idea behind it is we want to get local fishermen to be the ones who are actually cultivating the seaweed. We want this to be a technology that is really easy to take up for local people, just to at least get the market going, get the cultivation system starting to happen, and then we're going to be able to do more kind of technologically defined methods and things that are going to be a bit more complicated. But this is just a really simple system. There's a header rope here which is kind of weighted, so it sinks in the water a bit. We have our eco bags here, and we've got buoys along the top. And then our cedar and string is just kind of unwound around the top of the rope, and it's really as simple as that. There's not too much you have to do with it. You can put them out, just check them once a week, make sure they're okay. We do sampling on them. You don't have to do sampling, obviously, because it'll be done in our commercial systems. But it's really quite a simple process. We do a lot of outreach. We've done school talks, so we went out to schools at the start of the project to inform children about renewable energy and why it's important and why we're using seaweed. We figured getting in and getting to the kids was actually going to be a really good way of getting that information kind of filtered through to the parents, to the adults, in a much more relaxed fashion, instead of us trying to come in and push an agenda. If we go in through the schools and get them educated, then it'll filter up. It seems to have been working quite well. So myself and Emma, we took seaweed to schools, we did our little kind of demo of our blender, how we spread it, took a microscope so they could have a look at things, and they really actually did love it. We're sponsoring a local festival as well. So it's a maritime festival. It's run by the Strangford Lough and Lake Partnership, which is essentially a stakeholder supergroup. They're the spokesperson for all the local stakeholder groups in the area. So we got involved with them really early on at the start, and using the publicity and outreach funding that we get from the project, we're able to put that then into sponsoring the festival. And it's been really handy for us because we can essentially piggyback on what they're already organising and piggyback on all their publicity. And we just add in some extra activities, and it's worked really well for us. We've had a couple of thousand people come through the labs and open days. We've done boat trips, taking people out, they can have a look at it. We've had people eating the seaweed off the lines, and they've really, really enjoyed it. We do some videos, so we have videos up on the QML YouTube page. So we have our basic aquaculture videos, generally talking about the project and the biology behind aquaculture. We did an ITV program called Off the Beaten Track, and we were just like a little segment in it. It was like a magazine show kind of. And we have that video up there if anyone wants to see it. And we also have some footage and dive footage of our anchor rags as well, so you can see that there's seaweed attaching, there's crabs there, there's hermit crabs, there's loads of stuff. So it's not just what there, which obviously we have to do the data. So we take environmental data and biomass data. I'm going to present a little bit of the biomass stuff here. I don't really have an awful lot of time to go through everything, but we do monitor for nutrients, temperature, light, and we do benthic surveys to make sure we're not affecting the sediment, we're not going to be altering the system that's down there and having any negative effects. We're doing a hydrodynamic survey as well. We want to put down equipment that we can then monitor the current going through. We can have a look and see if you have seaweed longlines there, is that going to be affecting the current? Is it going to be affecting the water motion around the lines as well? Is that going to have an effect? And then the biomass analysis, so length, width, weight, wet weight, biomass composition, all the rest. So yes, it does grow. We got, and this is Saccharina from the first year. We've got some really good big seaweed. Ours is growing quite broad because it's relatively sheltered, so it works quite well for us. But we've had some really variable results. So this is year 1 data. So the number of plants kind of varies a little bit, partly due to our sampling. So whenever we go out, we sample 30 centimetre sections 5 times along each row. And we completely strip those 30 centimetres and take that back to the lab. But obviously each time you go out, you're sampling a different 30 centimetres, and then that's going to be having edge effects and affecting how things go around it. But it's the only really way we can do it. It does actually grow, so yes, the mass is increasing. We've had differences in some of our species. So we're cultivating Saccharina and Laminaria in the first year. The Laminaria is the blue and yellow bars here at the front. They really don't seem to like where they're located. They just haven't grown really well compared to Saccharina. Everything we take comes from the rock, so it's all local seaweed there. It's all evolved and developed to grow within that system, but just not in that exact area. It's quite a sandy seabed there. We do have some Saccharina there naturally, but we don't really have that much Laminaria there naturally, so I think it just doesn't like that area. I think it's just a bit too calm for it. This is the grid system now that we attempted last year. It really, truly, completely did not work. This is the kind of thing we're going to be putting into our best practices. Most of the guides out there will say how to grow seaweed. They don't really say how not to grow it. We're going to put a slight slant on it and include that, because we waste a lot of time whenever you're trying things that don't work. So the idea for this, instead of having seaweed just along these ropes here, was to seed in between, and then you can really kind of maximise the area that you're growing. In principle, it would be really good, except that we have these connections here. So they have buoys along here. It's really difficult to deploy it because you're constantly having to stop seeding and click on buoys, and you're moving the boat in and out. And also whenever the tide drops, the whole grid system, we couldn't actually place these anchors out here. So the whole grid system kind of collapsed in on itself like a concertina. And then whenever it was meant to come apart again, it possibly would have done, only the day after we deployed. We had a storm, there was a lot of driftweed, which they got tangled up, so it just wouldn't come apart. It just all stuck. So this is what happened. We had a buoy, rope was wrapped around the buoy, we had lots and lots and lots of driftweed that was wrapped in, and this one here you can see the string was actually coming separated from the line as well. This string should have been full of lovely seaweed plants, but it's as white as the day it was sprayed, so it really just didn't work. So we're going back to the old system for the next year. So this is some of the data from it. You can see here because we had the issues with the grid system, it obviously affected the growth. Alaria is a new one that we grew this year as well. It did grow, and the green is the biomass of plants, so it was increasing. But because of the issues, we lost a lot of plants. So the actual blue line here is the projected biomass, what we anticipated we'd be harvesting, and then it's decreasing because there's less plants. It doesn't matter if they're bigger, there's fewer. What we actually did harvest in the end is then a purple, and you can see it does not match up with what we would predict would be getting. So there's another issue here as well. The models are great, they're a great tool to have, but there's still a lot more work that needs to go into actually validating them and seeing just how realistic those models are actually going to be. We also tried doing some work standardising the seeding method. We kind of thought, maybe it's not just that we lost plants, maybe it was really patchy on the line, maybe the string wasn't spread really, really properly. But this is from the first year, whenever actually things did work really well. And you can see here, so the bottom bit is kind of the clumps. So they do tend to kind of clump together. So we had small numbers of clumps. this is numbers of individual plants at different lengths on a cedar string. There's just no pattern with it really. You tend to get a peak around here, and we think it's because whenever we spray, the bits at the top are kind of running down, and then they kind of stick around the middle, and the bits at the middle are running off the cedar altogether. So it tends to drop off down here, but it's still, there's no reason as to why this one just stays low and then increases way up here again either. So we've got a bit more work to do to see can we standardise that. If we can't standardise it, I think the best way is going to go to just try and spray as much stuff as possible onto the thing, and then whatever's going to grow is going to grow. So this is the system we're going to go with this year. We're going to have these 100-metre lines that are 3 in tandem, so it's essentially a 300-metre system. And then we're going to have a smaller grid which is based on what Jennifer has in SEVA. I presume you're going to be talking about that. This is going to be just a 50-metre wide system, and we're going to have 7 lines here, just as a small prototype. The idea is this, is that we can pack them closer together. So while we don't have the grid in between, they're packed closer together, so we still might be able to increase the productivity. No matter what we do, we still tend to get the same amount per metre. So the research that we're doing biomass, environmental data, benthic surveys. I'm going to speed up a little. Hydrodynamic stuff, biochemistry analysis. We have a new postdoc, Peter Sheener. He's going to be looking at the ID potential, the products that we've got, some basic to see what we have that we can actually sell commercially in our seaweeds. Is it going to be useful for something else, or just go straight to ID? What are we going to do with it? And we're also doing some genetics work to profile the seaweeds that we have to see are we going to be able to do either selective breeding for optimal characteristics, or are there going to be any interactions between wild and cultivated material as well. We're developing these little transport systems to make our cedars easier to transport for longer distances, and we're also looking at IMTA. So IMTA is a combination of different aquacultures. So you can have shellfish or finfish alongside seaweed. We're looking at it because it has environmental benefits. Seaweed as a bioremediation tool can clean up all the excess nutrients from fish farms or shellfish population. Seaweed is really expensive to grow still. It's just too expensive, and if you grow it in combination with something else, you can take your overall running cost down. You also have a backup crop as well, should you need it. Ocean is big, but it's not unlimited. Lots of people actually want to use it. You can't cultivate seaweed everywhere in the ocean, so it's not all up for grabs. The cultivable space is actually pretty limited as well. Diversion product, you get a risk reduction. You get a risk reduction, farms are more likely to back your business. You can tap into the market need easier with different products. Sustainability, which is the name of the game when it comes to this and algae project. If you combine products, you're going to make it much more sustainable. I'm not focused too much on this, it's just the general bioremediation, bioconversion, different ways we can treat our seaweed. We're not looking at all of it, we're trying to focus in on the biology and combination, but there's plenty we can do with it. And seaweed, the price of seaweed needs to come down a lot more because of what we use it for. It's simply too expensive at the moment, but we're getting there, it's definitely getting better. There's lots of things we can use it for. This was taken from the document that Brenda did with ADAPT, and you can see bioremediation is one of the most important things as a use for seaweed. That is an important thing to look for, and that ties into so much. It ties into IMTA, it ties into cleaning up water, it ties into CO2 uptake and everything else. So it's something that we're really looking for in the future. Thank you.
Thank you, Karen. Are there any questions for Karen? Yes.
Could you give an idea of production costs?
Production costs, to establish a hatchery, it's probably looking at around 130,000 euros, something like that, to set up a hatchery. Building a long line and deploying it is probably going to work out about €1,000 per long line. If you're looking at deployment and everything else, because you need a barge, you need a crane, you need to hire boats if you don't have those boats yourself. Like the actual long line systems themselves are cheap. Rope is cheap. Use concrete blocks, they're cheap. Our eco bags are expensive. But if they work as a stakeholder appeasement, and if they work in promoting the environmental diversity in the area, then it's something that we would be willing to invest in. exactly what it's going to be depending on what method you use, but you're looking at around about €200,000 for a pretty simple CD farm. So it's prohibitively expensive for small startups. The hatchery cost is one of the biggest things.
Okay. Your annual production, was it?
Pardon?
Your annual production?
Our annual production, well, because we're trying different things and a lot of the experimental systems don't actually work, you're still looking at around about 7 kilos a litre. You can probably get to 10. I've heard that number quoted quite a lot, but we still seem to be coming in around 7.
Just one more question.
Karen, at what point in that whole cycle do you get the greatest increase in biomass, greatest sort of productivity rate? Is it earlier on when they're really small, or is it later on?
I mean, whenever they're out in the lines, or
The whole process.
The whole thing?
I'm thinking here, could you actually take them out of the sea? So could, if could you just work with the small?
You can bulk up the seed in the hatchery whenever you're keeping them in a stasis phase, so they'll essentially just clone themselves. So it's a really easy way for us to take a small amount of fertile material and get loads of cultivable material after that. Once you're looking at putting them in the seed to get actual product that you can use, it's probably going to be a few months after they're deployed. So we deploy them about 2 months, 1 or 2 months after they've been in the hatchery, so they're still really small. They do jump. It's very much dependent on the environment that you've got. The Alaria, for example, can jump. It increased for us. We deployed in February. It was absolutely beautiful in around April, May. But then whenever, in June it was gorgeous. Once we went to harvest it in July, and it all died back, and loads of stuff had started kind of breaking down. So you're looking at about maybe May, June to harvest.
Short question.
Just luck.
What's the distance between two adjacent lines? And at
We
what
have-
depth are you cultivating?
They're 1 to 2 meters below the surface of the water, and we have them 10 meters apart at the moment. We're going to try with the small grid system, or the rat system we're going to use. We're going to have them about 5 meters apart. I think in Seva you have them 3 meters, 2. So you really can pack them in a lot more by doing that way.
Okay, thank you. Thank you, Karen. Moving on to the next speaker. This is Jennifer Champenois. She works for Seva in France, and her talk is about design and trial of a new seaweed production structure. The floor is yours.
Hello everybody, so I'm going to use the microphone. So I am Jennifer Champenois from Seva in France. We are involved in the Anarchy project as a seaweed pilot as well, like Queens. And before starting, I just want to thank my colleague Florian Fernandez, who has helped me a lot with this project for several months now. So today I'm going to talk to you about a new structure that we have designed for seaweed production at sea. What about our situation? So we are a demonstration pilot for seaweed production in the Anarchy project, and our current research focuses on how to improve the cost effectiveness of cultivation of kelp at sea. This is addressed by 2 aspects. First aspect is to increase the production of the biomass, which we expressed as kilos of biomass per meter of rope. And we are also looking to optimize the occupation of the area at sea. As you have realized with the presentation of Karen, we need to optimize the occupation of lines at sea to get the best yield expressed as kilos per hectare. So our facility in Seva includes an inland hatchery where we are producing plantlets on lines, and afterward the lines are transferred at our site at sea, which is a 6-hectare site with a depth comprised between 12 to 25 meters. It's a pretty sheltered area as well, with low wave action, but on the contrary, we have very high currents in our area. Earlier with Karen, you see that it was 0.3 meters per second. So at our site, at high tides period, it's up to 4 knots, which is more than 2 meters per second. So since the 19th in Seva, we were producing seaweeds with the traditional long line system, but because of the strong currents, we had to set up the lines 50 meters apart from each other to prevent the line from tangling, which will cause chafing and thus loss of biomass. That is why when you have to display such occupation at sea, you see here it was our occupation on the area last year with the lines 50 meters apart from each other. So even if you achieve the high productivity, the yield per hectare would be low anyway. That is why we wanted to develop a new structure to optimize the yield. So the idea was to set up a semi-rigid framework to strengthen the structure and to be able to reduce the space between the lines. So now we are working with lines displayed 2 meters from each other. We have designed 2 types of what we call production unit. So you see here our current occupation of the site. There are units of 50 meter longs and units of 100 meter longs. And we have currently 4 units at sea. So let me show you more in detail what is looking at. This is a scheme showing the general concept. This is not at scale. So basically the lines, which are in brown colors, are stretched between 2 big plastic tubes made of PEHD. And these tubes, they are anchored to bottom by big mooring system. There is big buoy, red buoy, to achieve the buoyancy of the whole structure. There are also some, in blue line you see what we call header ropes, which are larger ropes than the seaweed ropes, in fact. And these ropes help to keep the structure well settled, in fact. So we have also what we call a spacer. In fact, it's just the same tube than the main tubes, but they are smaller, and they are here to keep the space between the lines. Here you can see a cross-section view of our lines. So in dark, this is the tubes. The lines is kept at 1.5 meter below the surface by 5 little buoys, which are displayed 10 meters apart from each other. And this is at scale views now, so at the top it's a scheme, and at the bottom it's pictures taken at sea. So some colleagues had the opportunity to see the site last May, and here it's the 50-meter unit. So I have to say that this new structure has been designed in collaboration with experts involved in design of aquaculture farm dimensioning. This is a specific result of their study about the mooring line design. And according to their recommendation, this is what we actually set up at sea. So you see the main tube is attached to the red buoy at 3 points. There is also an underwater buoy which helps to keep the rope here and the chain well stretched at whenever, depending on the tide. It helps to prevent the chain not to friction the bottom of the sea. And at the end, there is the mooring system with 2 concrete blocks, one of 3.5 tons, one of 2 tons, and an additional anchor made of steel. It's a 200 kilos anchor. The total cost for this new structure was 40,000 euros. So this includes the feasibility study, all the tubes and the anchoring equipment as well, and also the manpower for setting the equipment at sea. For example, you need big boat like that to set the anchors at sea. This is not including the seeded lines, okay? It's only equipment. The pros and cons of this new structure. Starting with the pros, so we are expecting a higher yield per hectare at the end. There will be less moorings to survey for maintenance operation because every year we have to check if everything is alright with the anchors. Regarding the cons, obviously it's a structure with a very strong tension, so we have to keep careful. And a second cons is that you need a well-adapted boat to be able to navigate within the structure, and especially for the monitoring of line that we are doing during the algae monitoring, we have to be able to go above the lines to see if everything is growing well. But for harvest, it would not be a big problem because you can imagine to harvest the line by pulling them away from one side. So you don't have to be within the structure. So we have set up the unit at sea in the fall 2013, and afterward we have run a trial. So let's see the cultivation trial now. We were looking at 2 species, Alaria esculenta and Saccharina latissima. We, for cost reasons and labor cost reasons, are using the seeding technique, which is the direct seeding. So we are using spores. We don't go through gametophyte cultivation. We go directly with spores. The spores are settled onto strings that we kept in hatchery for 4 to 5 weeks. And after, we coiled the string around a larger rope and put the rope at sea. So you see that for this specific trial with Alaria, the fertile material was coming from the wild, a place called Ingrande. We seed the lines with 2 different densities. The seeding happened in the 4th of December, and we transferred the line in the 14th of January. For Saccharina, the same. The release of spores was done from wild fertile material. We again tried 2 different seeding densities. Date of seeding was the 5th of November, and transfer of lines at sea, it was the 11th of December. So, as Karen has explained, we are doing monthly monitoring within the NRG project, and we have agreed to do the same way. So, each month at our place, it's from March to June, basically. We are sampling 5 different pieces over the lines, and we are doing different measurements on these samples. So we are looking at productivity, expressed as kilos of wet biomass per meter of rope. We look at density of plants, morphological characteristic, and also drying matter and biochemical composition. We also monitor environmental data. What is great is that we have the same sensor between the sites, so we can compare easily the data that we produce. So here you can see the data regarding underwater light intensity and underwater temperature. It regards the period going from November to June, which is the period when the lines were at sea last season. An important fact is that at our place, the temperature is very constant over that period, from 10 to 13 degrees, which is perfect for the growth of carp. We do also monitor the concentration of nutrients at sea, especially the nitrogen and phosphorus. What you can see is that for nitrogen, it's much more available during winter time. But this is very rough assessment of seasonal variation, because regarding nutrients, we are sampling seawater samples only once a month, so it's a very punctual monitoring. So let's see the results now. Here you can see the density of plants for both species. SL is for Saccharina, AE is for Alaria. And we have the 2 different densities for both species. So what we see is that in most cases, there is a decrease in the density of plants over the months. In fact, at the beginning, there are many tiny plants, but they are competing for light and nutrients. So over the time, some will take advantage over the others, and that's why you have, in the end, a natural clearing of the population along the rope. This was not the case for this line, which is the Alaria line, which was seeded with the lowest density, and it's probably the fact that there were less plants at the beginning, so the competition was less important. Anyway, at the end, we see no difference between the 2 seeding densities that we have tried. We achieved a density of more than 500 plants per meter for Saccharina and more than 200 plants per meter for Alaria. In terms of productivity, so it's kilos of biomass, wet biomass per meter of rope, we see an increase over time. So, okay, everything is growing well, that's good. And there's no difference between the 2 seeding density. For both species, so it's the same. And we have a higher productivity for Saccharina, so we reached more than 20 kilo per meter for Saccharina this year at the end, and it was only like 10 kilos per meter for Alaria. So this might be the fact that we see the biomass with less spores, but in more general approach, what we already see is that for Alaria, it seems that the species prefer more exposed sites. This comes from previous trials that we were carrying at different sites around Brittany. We have seen that the sites which were exposed give better result for Alaria, but as our site is pretty sheltered, then we can say that, okay, it's not so good for Alaria. In terms of morphological characteristic, a funny fact this year is that with this lowest density, conclude. Okay, so
A couple of minutes.
okay. So lower density was used for alaria, and what we achieved is wider alaria. So probably less competition between the plants, and then we get larger plants. And it was interesting for people involved in food processing, for example. That was they told us. As Karen says, we have epiphyte at the end of the cultivation period. So at our site, basically, we have to harvest before June. So April, May appears to be the good time to harvest at our place. Whereas you have a loss of biomass and also a loss of quality. You can also decide to harvest at one day or another based on variation of the composition. As you may know, there is seasonal variation of the composition. For example, here is the result for Saccharina. So if you wait for June, there will be much more mannitol in the biomass. But if you want a biomass which is enriched in protein, it's okay, you can harvest in April. Okay, so I'm going to skip that part. We will try again with the new structure because we are very happy with the results. We will extend to 4 species, Alaria saccharina plus and 2 laminaria species because there is a demand on the French market. And we will compare 2 seeding density, direct seeding on strings, and also direct seeding of, on ropes, which will help to reduce labor costs, in fact. We can discuss about that later if you want. And again, but it will be fast as Karen explained about the IMTA concept. We are very happy to say that we finally get our license and we are moving to IMTA now. So it was the good news, it was in this very summer. So we are looking to develop an IMTA pilot farm now. And this is our plan. You see, there will be the seaweed unit, there will be mussels line, and fish cages. So we can go into it, and we are open to any collaboration. Okay, so thank you.
Thank you, Jennifer. Any questions for Jennifer?
Hi, Jennifer. You were saying from your graphs that your sort of maximum biomass was around June. What's actually the biochemical composition doing? You didn't show data. I mean, I was working in a similar area before, sort of looking at carbohydrates, and actually what we found was that sort of the maximum